My name is Astha Symes, and welcome to Live Longer World. I'm really excited for this conversation because I think you bring such a multifaceted approach to looking at cancer from mathematical modeling, game theory, and also evolutionary lens. So, just to jump in, I would love it if you could discuss the new framing of the hallmarks of cancer, not as a static process, but one which you see from an evolutionary lens. So, you've discussed how the tumor gets initiated, it changes its tumor microenvironment. And then you also start to see this emergent phenomena of cooperation among these tumor cells, something which Mike talks about a bit as well. How do you see this evolutionary progression of cancer?
Well, um, from an evolutionary point of view, cancer is a fundamental evolutionary event, and whenever you talk about evolution, you talk about what's the unit of selection, and in for our body, for the cells in our body, the unit of selection is us. Our cells have the same fitness as the three-dimensional multicellular organ, us. Um, and they share that with us. They contribute to our fitness. And so they're controlled by the structures of our tissue, the local tissue instructions. Their death, their proliferation, their movement, their phenotype, all that is controlled by the tissue, by tissue signals. And as a result of that, they do not evolve. Um, Whereas in cancer, it's the unit of selection is the individual cell. So that carcinogenesis means that there's a fundamental transition of cells that are defined by the fitness of their organism to cells that are individually subject to evolutionary selection. And that's a tremendously, I think, important way to look at this. The traditional view of carcinogenesis is that it's an accumulating mutations. Something about those mutations generate a cancer. Um, in this setting, what we would say is that the cancer cells have to become independent of the host tissue signals. Um, one of the problems with this kind of sequential mutation model is observations that you can see the same mutations in the apparently normal tissue adjacent to the cancer, which seems to suggest that's not, that it's a part of it, but it's not necessarily the cause of it. And so in the evolution model, we admit that you may get these accumulating mutations, which renders the cell, blind and deaf to the tissue signals. So they become totally independent because they just can't receive them. On the other hand, an alternative is that you can damage the normal tissue, and that damage prevents it from controlling the local cell population, which now allows them to evolve. And now the accumulated mutations that have occurred over a lifetime, which are similar to, all around, but now these mutations can become part of the fitness function of an individual cell. So there's sort of a subtle difference in this, um, and so it's that transition to independence that's the critical function. And what this suggests, I think, is that cancer is not just a mutation. It just isn't happening to the cells. It's happening to the tissue. So, damages to the tissue, inflammation, injury, or just old age, where the tissues sort of lose control of the local cell population, can then trigger this evolutionary process. And so when you think then about cancer as an evolutionary process, the hallmarks of cancer then need to be framed in terms of how they add to the fitness of the organism. And so one of the things that organisms do is called niche construction. Beaver dam being the most obvious one, but that's kind of been added to evolutionary theory over the last decade or so as part um, of the inheritance process, because the beaver dam is an example again, can last for several generations. And so it can persist as an ecological mechanism of inheritance. And so, when you start to think in these terms, um, to me, a great example is the Warburg effect. And this has been known since Warburg, I don't know, it was 100 something years ago, that cancer cells, even in the presence of oxygen, prefer glycolytic metabolism, meaning that they don't use oxygen to metabolize glucose, they just metabolize it to lactic acid. And the problem with that is that it's very inefficient compared to aerobic glycolysis or aerobic metabolism. And so that's often called a dysregulation. There's something wrong with the cancer, that it's not doing what it should be doing. And, evolution doesn't do dysregulation. Evolution is, the greatest optimizing force in the universe. So if you're seeing the cancer cells use glycolytic metabolism in the presence of oxygen, that means it's conferring a fitness advantage. Um, and my own hypothesis is it's the acid that it's produced that's the fitness advantage. It's a niche construction, so that the cancer cells now can adapt to a high level of acidity, which normal cells cannot. It promotes invasion, and it tends to blunt the immune response. And so it serves a purpose. Um, and I guess that's the kind of the theme here, is that evolution is a scientific game of Jeopardy. Evolution gives you the answer. Our job is to provide the question. Evolution tells you that aerobic glycolysis is necessary. It increases fitness. Our job is then to find out why. It is not to say that evolution is making an error. That just will not happen. And so the idea that this aerobic glycolysis is a dysregulation, is an error in metabolism, and that somehow is at the root of cancer, is really kind of missing the point. And it's interesting to talk to pathologists who use stains to identify tumor cells, PSMA stain in prostate cancer, for example. Um, you can then ask, why is the PSMA overexpressed? And it's funny because it becomes a circular answer. It's because prostate cancer cells make it, and that's how we identify them. And I have to say that no, it's not there for you. It's there because it's increasing the fitness of the cancer cell. Your job is to understand then why PSMA is increasing the fitness. It's nice to say that you can use that as a diagnostic tool. GATA3 increased expression in as a marker for breast cancer, but it's also evolution telling you this is really important, and that connection is often not made, I think.
Interesting. So instead of seeing the hallmarks of cancer as static, dysregulated traits, you instead see them as traits that evolve to enhance the fitness of the cancer cells in order to allow them to spread further.
Yes, this is evolution's message to us. Evolution is telling us this is what's important for cancer. This is what's necessary to optimize its fitness, which means to optimize its proliferation. We should listen to evolution more than we do. Um, we talk about mutations, and clearly, if you see mutations in a lot of the cancer cells, then that mutation is optimizing fitness. But the opposite is also true. If you don't see mutations in the cancer cell in certain genes, those genes are necessary for fitness. And although we always talk about drugging, mutations, I think, and we have some math models that have looked at this, is that actually targeting the genes that are conserved is probably a more effective way to treat. than the mutations, because those are unconditionally necessary for optimal cancer cell fitness.
Why do you say that? What does it mean to treat the genes that are not affected?
So what that tells you is if you assume that all the genes are undergoing random mutations, they then undergo what we've called evolutionary triage, meaning that if they increase fitness, the cell will proliferate. If they decrease fitness, the cell will not proliferate. So over time, what you'll see is that any mutation in that gene is selected out, meaning that evolution is telling you that a mutation in that gene decreases fitness. Evolution does not want that. That gene has to work perfectly. And so attacking that gene is attacking something that's very important to the cancer.
Right, I see. Okay. This maybe does this feed into some of the atavistic theory of cancer as well, in terms of attacking some of the weaknesses of cancer instead of the strengths of cancer?
I think it does. The atavistic theory is that cancer sort of becomes more primitive, and there's something slightly pejorative about that, way of looking at it. Oh, they're primitive cells. In fact, they've evolved to a very high state of fitness. Um, so, so I sort of admire them. I mean, they're deadly but beautiful evolutionary processes. They can basically withstand almost anything we can throw at them. Um, so atavistics feels like it's like it's we're minimizing the complexity of cancers.
Taking this fitness function forward, is it fair to view that, say, in normal cells, there's a degree of cooperation, right, for them to function? And then the cancer cell, it becomes this individual cell that starts off as an individual cell, and then it adapts these functions to gain cooperation again, but it's a different level of cooperation now at this point.
Yeah, the word cooperation is so anthropomorphic that it's I think you have to be careful about using that. The evolutionary biologists tend to use the word mutualism, or something like herd effect. So, for example, herd effects being, I guess, the most obvious one, where these animals individually are the unit of selection, but groups of animals can act in a way that protects them all. And so groups of cancer cells need to have at least a loose affiliation to work. So, for example, the cancer cells need blood vessels, but it's probable that one cancer cell cannot produce enough androgenic, molecules to bring those in. So the whole group has to do it. Now then there's cheating that can occur. So you can, a cell in this group that's getting blood vessels in can decide not to make VEGF, for example. And so now it's getting the benefit of the group, but not giving not paying its due cost. And so these can be very complicated dynamics. But the word cooperation implies sentience, that there's some thinking that's going on. And I think I tend to like to avoid that. One of the things that I've noticed in working with oncologists, is that there's almost a tendency to think magically about the cancer, that somehow it's these are plotting against you, and they has a remarkable capacity to overcome your therapy, and so there must be some inherent evil in it, and I think it's more mundane than that, but it's not playing. They're not sitting down there, like, gee, how can I get around this? But they are extraordinarily powerful because they have the whole human genome to bring to bear on whatever evolutionary problem they face.
Mike, you often talk about cancer as dissociating from the collective behavior of cells or the tumor cells. So, what are your thoughts here with the herd behavior and evolutionary processes?
Well, I have a couple of things to talk about. I think one thing I'd love to dig into a little more is the notion of stress initiating these processes. And there's a particular kind, and so you mentioned tissue level stress, and so one of my favorite examples from our developmental biology world is this old finding that when you take a tail of a newt and you graft it to the flank to the side, the thing turns into a limb. And if you look at the cells at the very end of the tail, they're tail tip cells sitting at the end of a tail. There's nothing locally wrong. Nobody's poisoned. Nobody's damaged. And yet they completely remodel this. The issue isn't at the cell level. It's at a much higher level that then filters down and makes the molecular biology dance to this higher level error. So my question is, what typically do you think the cells are measuring that leads them to be unhappy enough to break the multicellularity contract and say, you know what, this isn't working out for me. I'm going on my own. Like, what is it? Is it metabolic? Is it like what usually triggers this stuff? Do we know?
Well, it's a great question because I'm not really sure. I mean, and we've talked about this a little bit. I mean, there's this tendency to think about diffusing hormones and diffusing signaling molecules and receptors and things. That seems to be nowhere near enough to get the level of complexity that you see in cells. I mean, they in tissue. I mean, it's their location, it's their phenotype. I mean, there's this extraordinary synchronization of all this. I tend to think that, and Mike, we've talked about this, that actually we're missing a key information dynamic in cells, and that is I guess you would probably call it electricity. I would maybe say ions, that there's communication that goes on that's not genetic. And that is fundamentally important. I think it was, I don't know if you remember this era, but when the human genome was being deciphered, there was a contest, and this was around the world. Scientists from all over the world could put in, they would bet. It was a pool, and in fact, I think it was called the gene pool. And they put in, each of them put in, I don't know, $20, and they bet what the number of how many genes would be in the human genome. And 400 or 500 scientists across the world took part in this, and basically none of them were right. They measure the mean estimate was about, I think, 60, 70,000 genes. Upper ranges were in the 300,000. The lowest was 27,000, and the number is it's under 20,000. And so David Baltimore wrote an editorial with in that, I think it was published in Nature, and said, we need to understand this. You know, if we believe that we are the most complex of the organisms on Earth, why is our genome smaller than that of a mouse? And I think the argument I would have is that it's because the genome only indirectly controls the information dynamics. It sets up the dynamics that are based on ion conduction and on electricity flow, that really are at the basis of complexity and of these interactions among cells. And one of what to me is interesting is that our cells have tremendous transmembrane ion gradients. They're enormous, and mammalian cells spend about a third of their energy budget maintaining these, which is about as much as they use maintaining the genome. Now, If you remember the Watergate thing, you follow the money. And I think in evolution, you follow the energy. What evolution is telling you is that this transmembrane ion gradient is really important. It's so important that it devotes a tremendous fraction of its energy budget, and yet we know no reason for that. I mean, it serves no apparent purpose in the current sort of understanding of biology. And what I think is that that's a tremendous um, a gradient of information, and cells interact with each other, opening ion pores, which allows flux of ions across these gradients, and those fluxes are information. And that another interesting, factoid is that elements of the cytoskeleton uh, are tremendous ion conductors. I mean, they're really good. I mean, they're described as living wires. Um, why is that the case? And again, there should be a purpose to that. Evolution doesn't do things with no purpose. And I think that is sort of a kind of wiring that allows ion flows across the cell to communicate within the cell. And then I think Mike does a lot of work with how cells communicate among themselves, all of which is through ions, conductivity and stuff. There's no genetics in that. The genes set up the dynamics that allows, they make the pumps, they make the pores, and this is necessary for this other information dynamic, but I think it's an information dynamic that has been sort of missed over the years, or at least it's hinted at, but not, or let's say it's not necessarily in the mainstream of biology. So I think that to understand multicellularity, and complexity, I think we need to invoke a different information dynamic. Um, going beyond the genome.
Yeah, for sure. Do you want to talk a little bit about your recent paper on the computations in the networks within cells? I thought was incredibly interesting. Do you want to talk about that a little bit?
Well, it really goes to that idea is that, and I don't know if it may be going off the deep end here, but there's something called Maxwell's demon. And if you're familiar with that, Maxwell proposed this Gedanken, I mean, a thought experiment. This was in the mid to late 19th century, and is when thermodynamics and Boltzmann kinetics were being discovered sort of simultaneously. And what he said was that suppose you have two boxes with gases at the same temperature, and there's a hole between them with a gate, and a demon sits there, and because the temperature, because in any given temperature you have this Boltzmann distribution of velocities, and what he said, the demon would open the gate if a high velocity molecule was coming toward it, and close it otherwise. So essentially what he did was to shift all of the high velocity molecules to one box and the low velocity molecules to the other. And basically, so this one box would be hotter than the other. It would look like a spontaneous flow of heat in isothermal between two systems of the same temperature. It's also a spontaneous reduction in entropy. And he said he viewed this as a violation of the second law of thermodynamics. And it's been controversial ever since. But what it gave rise to in the early 20th century is information theory, because what they said is the demon has information. And so that's become sort of a link. And there's so the ion gradient across the cell membrane is similar to what the demon does, but with velocities. And what was really never appreciated is the fact that once the demon has set up this gradient, you can have Maxwell's angels that can sit on the looking out for information coming from the environment, and they can open a gate. And now ions can flow down this gate according to the gradient, and that is a flux of information. And so I think that's an important part of how cells interrogate their environment. And now the gross part of this so there's this molecular level difference. These are the ion gradients, but they converge to make a transmembrane potential. And this is at the border of ions, and so you have this molecular level dynamic, and you have this bigger scale dynamic. And for Maxwell's demon, it was really the gas molecules and as changing the Boltzmann kinetics of each of the chambers, but manifesting as a thermodynamic change. That I think that intersection of microstates and macrostates is kind of where information works. In this case, the information from the genome pumps the ions against the gradient using energy. Again, there's got to be a reason for that, and I think you can calculate the amount of information within that by Shannon information criteria, and it's enormous. It's orders of magnitude greater than the information content of the genome.
Yeah. Another interesting thing that you were mentioning before about aging, and we have this new, so I think it might be relevant to this new work where we look at morphogenesis as a kind of a homeostatic process which tries to minimize error from a certain set point. And we did this simulation where we looked at what happens to this goal-seeking system in the cybernetic sense, right? What happens to that system after it has completed its goal? What does it do after that? And what we found is that in the absence of any additional noise, thermodynamic, damage, no entropic stuff at all, even without all of that stuff, what the morphogenetic system does is it reaches its goal and it builds a nice pattern. So there's your embryo, and then it hangs out for a while, and then the cells start to disband. The cells start to go off and do different things, because that pressure to keep to upkeep a specific morphology is no longer there. They've sort of done it, and now what? Mission accomplished. And it, it's sort of my a scheme that I've been developing on this idea that the goals are what keep collective intelligences together, and that after that goal is no longer active, they just start to go off and do they go their separate ways in the absence of any external damage. It's a fundamentally a cognitive problem. It's not a thermodynamic problem, and even there. And then you start thinking, well, what happens to how do immortal animals deal with this? Well, planaria, who are both extremely cancer resistant and ageless and highly regenerative, they tear themselves in half every 2 weeks, and they sort of refresh this thing. They don't have a chance to sort of settle down and wonder what to do next, because they have this radical damage event that they cause themselves every couple of weeks, and that seems to be enough to keep them rolling. And so I just wonder, this in the connection between aging and cancer and everything, that's what do you think as part like a stressor can be? And we all know people who they retire and they don't know what to do, and then the things go poorly after that, right? So it seems like that's a whole other new kind of stress. It's not like there's anything happening to you, it's that the goal setting is kind of gone.
Yeah. Maybe that's where exercise is creating these sort of micro stresses.
Yeah.
Keeps us alert, keeps your cells in the game.
Yeah.
But one of the interesting things that I've been sort of thinking about is that the information in a gene is translated into a string of amino acids that can do precisely nothing until it folds. But that folding process is it's almost a probability matrix. As it can fold into a number of different configurations. And because of that, there's actually information gain in the folding process. And I'm not sure we've sort of understood that. But what's interesting then is that proteins that form enzymes, for example, are fine graining because they act at a quantum level. But what they do is the substrate have the quantum interactions of the substrate need to be optimized to make the enzyme work. And fine graining requires information. And it's occurred to me that the way life does this is the protein folding, that going from one dimension to three dimensions in a sort of predictable way provides an information gain. And I wonder if some of multicellular organisms have that same kind of process where the cells, you have essentially a point source in the sense that a cell is a point source. As it proliferates, the interactions among the different cells are sufficiently predictable. Somehow in this fertilized egg, the information content is then sort of magnified, is increased by these interactions, these non-random distributions of the cells, so that the collective information of a multicellular organism, which is vastly greater than that of a single cell, is gained by these dimensionality changes. It's kind of hard to explain that, I guess, but it feels like biological information is not as simple, I think, as everybody says, oh, the biological information is in the genome. So great. How do you go from genetic information to the thermodynamic state of the cell, which is a highly highly ordered, low entropy, but far from thermodynamic equilibrium state? It's extraordinary. There's nothing in the universe like that. Intuitively, it seems clear that genetic information does that. But to me, it's not clear how you go from genetic information quantified incidentally by bits. So there's no physical unit in that, to thermodynamics, which is of course quantified by the typical units of thermodynamics. You don't have unit matching in this.
Yeah. I mean, there is some theory around what bits cost, like the minimal cost of a bit, right? The Bennett stuff and all, Landauer and all that. But I think, I think the bigger issue with the genome is that it's information about the hardware, and as you pointed out, it's some very cool reprogrammable hardware that it encodes, and after that, there's only so much you can blame on the hardware itself. There's a lot of physiological dynamics afterwards that are the main show, I think.
Yeah, it's magnificent. It's an extraordinary thing.
Yeah.
And as I said, I think I've become interested in the idea of dimensionality. The linear string of the DNA folding into a three-dimensional configuration. A single sort of point source as a fertilized egg turning into a large three-dimensional structure. There's those rules may be similar, I think, and the changing dimensionality may have it may be where how life exploits the limited information that comes from the genome to generate these highly large and complex organisms.
Yeah, and that's just in 3D like in 3D space, right? You also get structures in physiological state space and transcriptional state space, kind of,
Absolutely.
metabolic, like who knows? Like there are
Sure.
all these different spaces, right, that it's
Absolutely.
unfolding into.
And then, I mean, to get like really wild, you could say, well, what's the human mind, which is also sort of unique in nature. You could argue, I think, that what evolution is going to select for things that allow you to live longer, or to survive, or to proliferate. There's no evolutionary selection, to figure out the stars, or to, engage in quantum mechanics. There's no, it exceeds what evolution would have selected. And, my kind of armchair and speculation, which I'll deny ever saying, including saying that this whole broadcast was a hoax, is that we have we recognize an additional dimension, time, that our ability to work in the dimension of time adds a dimensionality change, which allows us to access information that would not ordinarily be present in strictly three-dimensional structures. Again, I'll deny I ever said that.
Going back to information dynamics, I guess how do you think about information dynamics for a cancer cell then compared to a healthy cell?
Well, I think there's two components to this. The healthy cells, are really tuned into their environment, into the other cells in the environment. They're tuned into this collective. I think Mike used the term. Cancer cells have to be independent of these control mechanisms. At the same time, cancer cells have to be able to forage. They have to be able to detect sources of substrate, and they also have to detect bad environments, and probably ideally they have to detect predators like the immune system. So the information dynamics change, and that's where, I think that calling them atavistic is sort of insulting to them. They are extremely fine-tuned to optimize their proliferation in the middle of your body. And and that's often not a very good place to be. I mean, some of them, they grow in the lymph nodes, where it's that's like robbing from a police station. The whole immune system is around them, and yet they continue to grow. So they need sophisticated information dynamics, but these are different from what I think normal mammalian cells have. But what's I think is really important to cancer is that the normal mammalian cells, a lot of the genome is blocked. They don't have access to a lot of the gene. Cancer seems to be able to access the whole human genome. So they find things that were the genes that were being expressed in the fetus that just weren't expressed in adults, but they can use that. So they find ways to evolve, and to optimize their fitness using parts of the genome that ordinarily are not accessible. And I think it's remarkably clever, um, and really scary, because they're really good at it.
But I suppose from an evolutionary perspective, I mean, evolution would obviously want us to reproduce and live as long and pass on our genes. So the information that it would pass to us would be the ones that normal healthy cells have. Where do you think cancer cells are acquiring this information per se? Or do you say maybe it's the fact that evolution has just programmed us for fitness and cancer cells just have this ability to figure out whatever is needed in order to proliferate?
Well, I think, after you've reproduced, and I'm sorry, I know you have a new baby at home. But after you've reproduced, nature would rather you were dead. I mean, and of course there's the whole grandmother hypothesis and various things that are that produce that increase fitness of offspring. But basically, if you're a man at my age, evolution would rather I just drop dead because in fact, that's why I exercise, because I'm trying to convince my body that I'm still part of the hunt, and it'll let me live. But I think that's a really important point. There's no evolutionary selection for anti-cancer mechanisms beyond the sort of reproductive age, and we live far longer than our reproductive age, unlike I think most animals. And so there's unfortunately no selection for people that get cancer in their 60s and 70s. It's just because we've reproduced. So evolution is pretty harsh.
But if you think about it from an information standpoint, because cancer cells also have some information embedded in them in terms of just how they want to behave. So do you think after we've reproduced, the information dynamics change in the sense that the information that the evolution programmed us for being healthy just shifts, or it just no longer cares about that? And I guess what I'm trying to understand is how cancer cells acquire this different set of information dynamics.
Yeah, I think that the James DeGregory is a great scientist in Colorado who looks at aging. And one of the things that happens with aging is that there seems to be a loosening of the tissue controls over the individual cells. And so probably that contributes to the fact that it's a process that occurs over generally in older adults. And I think the information dynamic. I don't think the information cancer uses was not necessarily information that was there for the cancer's benefit. It was there because it benefited fetal cells, or there's some part of it that's that the cancer cell can then adapt. And of course, it's not just the gene itself, but there's the spliceosome. There's there's a number of things that the cancer cell can change. Not to mention the role of the environment of acidosis, things like that. It can change the environment in ways that are beneficial. So, but I will not pretend to know all of the details of this.
Do you have any I mean, you must get this way more than I do even, but I get, we publish some things on cancer, and we have a lot of patients writing to us, saying what to do. So do you have any, and if you don't, that's fine. Aside from sort of follow whatever your best oncologist is telling you, do you have any other favorite thoughts for people to look into, whether that be like Seyfried's, metabolic stuff, or anything else at all? Do you have anything that you tell people?
As a cancer prevention agent, is that what
Well, I think beyond that. So once somebody's already been diagnosed, any suggestions there?
Well, cancer is an evolutionary process. Cancers, like all living systems, have to obey the laws of evolution. And so cancer is not magic. It's not an evil entity. It's just an evolving population. Now we know evolution. I mean, we know the rules of evolution. And we can use those to optimize cancer therapy. So, for example, the standard practice in oncology, which has been the case for 5 decades, is that any therapy is given at maximum tolerated dose continuously until progression. It's almost a mantra that they have. Evolutionarily, you could hardly do it worse, because what you're doing is that you're placing strong selection pressure for resistance on a large and diverse population. And you're also removing their competitors. You're killing the cells that are sensitive. You're selecting the ones that are resistant, and these resistant cells are now open they have no competitors. And so it's open for them to proliferate. And this is well known. This is actually a phenomenon in that's observed in evolution that's called competitive release. And this is not this is a strategy that optimizes the proliferation of resistant cells. So one of the things that we've tried to do is to bring evolutionary dynamics into cancer therapy. So, for example, we've used something that's called adaptive therapy. And here, what you do is, instead of giving maximum tolerated dose continuously, what you give is cancer dose. I mean, you give a drug dose a little bit. In prostate cancer, for example, we use the serum, the biomarker, which is PSA, and just drop it 50%. And then you stop there, you withdraw it. The tumor comes back, but the tumor is now proliferating with no selection for resistance. And because in general, in the absence of therapy, the resistant guys have some molecular machinery that allows them to be resistant, which is really good when you've got, it gives them a fitness advantage when therapy is present. But in the absence of therapy, it's a cost with no benefit. And so in general, the sensitive cells are fitter than the resistant cells in the absence of treatment. So the tumor comes back, and it recapitulates the distribution of sensitive and resistant cells that was there at the beginning, and you just treat it again. You just keep pushing it down. Now, this is in metastatic cancers that are fatal. This is where the goal is to maintain life with as highest quality as possible for as long as you can. So what we've learned is that we can in this kind of mechanism, we're only giving about half the drug that you would ordinarily get. So the side effects and the costs of the drug are much less, but we can prolong life considerably with that kind of approach. It doesn't have the sort of sexiness of trying to kill all the cancer cells. I mean, that intuitively is what we would like to do. I mean, that just feels very appealing. And that would be the right thing to do if you could. But given the fact that we don't have magic bullets, we don't have treatments that will kill all the cancer cells. And standard, a good example is androgen deprivation therapy for metastatic prostate cancer. It's really effective. 90-95% of the time, it reduces the PSA to normal or even unmeasurable, but it's also never curative. I mean, we know that just historically, it just is never curative. So, that's the dynamic you have. And the goal is then, to reduce the amount of ADT that you give, androgen deprivation therapy, which men hate, by the way, and it's got a lot of toxicity, significantly decreases quality of life, and typically gives you maybe 3 years. But if we can do much better than that so that they have a normal testosterone level about half the time, we've both reduced the cost and we've reduced the toxicity while also increasing, lifespan. So that's kind of the approach that we've tried to take. And so that's worked quite well. But it's so different from standard cancer therapy that it's really kind of intuitively unappealing to oncologists and sometimes the patients, because you drop the PSA to 50% of its original pre-treatment value, and there's something that says, let's just keep hitting it, that's the right thing to do. So it takes a sort of you have to think through that and overcome that kind of that intuitive sense that you're not doing it right.
And is that sort of thing just in terms of availability? It's so you guys are doing it at Moffitt. Is that it, or is this accessible to people, or like how is this looking now?
It's there are there's large trials going on in Europe and Australia. There's one using this approach in ovarian cancer that's going on, and is a multi-institutional study in the UK. There are some physicians, oncologists who are willing to do this, but it would still be considered, I think, non-standard practice. And so it will take a long time to I think change ideas about this.
Yeah.
If you were to hypothesize, where do you think adaptive therapy might not work the best?
We don't know. I don't think we understand immunotherapy sufficiently well. Now, the basic rules should be the same. I mean, evolution is evolution. But what we don't know are we have like we have the cancer cell population, which can evolve, and we have the immune response, which can change. And those are very complex dynamics. And we're just beginning to sort of look at those dynamics as they're very complicated, and there's so many things that are going on. It's difficult to put all that together. It's much easier to work with hormonal therapy and chemotherapy. Targeted therapy seems to be perhaps slightly different. But I will continue to argue that fundamentally evolution are first principles. We evolutionary dynamics will hold. These principles will occur. It's simply we don't know how they play out in some of these treatments. But I think you can argue. I mean, when I first was working at a cancer center, which is a long time ago, there just weren't effective therapies for a lot of cancers: lung cancer, renal cancer, melanoma. I mean, there's just nothing. And what we have now is really pretty good therapies for almost every cancer. And yet metastatic lung cancer is fatal now as it was then. And the reason is evolution, that the cancer cells evolve resistance to an initially effective therapy. And so I think increasingly you could argue that evolution is the proximate cause of death in cancer patients. And I think unless we acknowledge that, and unless we start to really actively integrate evolutionary principles into our therapy, we're not going to necessarily make progress. And unless we find a magic bullet, which may or may not exist.
Yeah, I'm curious, what are some of the open questions you're tackling now by integrating more of the evolutionary dynamics into cancer?
One of the things that we need desperately are biomarkers to understand intratumoral evolution over time. We don't have a good way to estimate that. Recently, and sometimes you just need more data, just need to acquire more data. A lot of our trials have been done on shoestring budgets, so we didn't necessarily have the ability to do some of the tests that we would like to do. But something as simple, what we found recently is if you take the testosterone level and the PSA level simultaneously, the ratio of that can tell you a lot about the subpopulations. So it doesn't have to be esoteric, circulating DNA. We can use kind of standard tests by understanding the connection between them. So what we find is if the PSA should reflect the testosterone level in general, but if you start to see the PSA for any given testosterone level, if the PSA starts to be increasing over time for that individual, that means there are cells present that are able to use much lower amounts of testosterone, much lower concentration of testosterone to proliferate. And so they're making the PSA, but the testosterone level is low. So something like that then allows us to understand that there's this population of resistant cells is increasing, and we need to do something about that.
Interesting.
I know you've also discussed this idea of habitat imaging and looking at the tumor microenvironment itself and then personalizing protocols based on that. One, I'm curious how that personalization gets done, and then two, I guess, why is this something that's not being done right now?
Um, the if I don't know if you've ever seen habitat maps. I'm sorry, species maps. So if you look at Florida, for example, and say, what's the distribution of ground squirrels? They'll be, they'll show you maps of where the gray squirrel is dominant, where the fox squirrel is dominant. Um, People don't walk around, every square meter of Florida measuring squirrels. And the way it's done is what's called landscape ecology, and this was developed for satellite images and basically large spatial scale images. By identifying habitats, you can then and then investigating a habitat and counting the squirrels in that one habitat, you can then do a distribution. So, for example, gray squirrels are very good at foraging, and so they can outcompete the red squirrels and the fox squirrels for competing. So if you see a college campus, you will basically always see gray squirrels there. What fox squirrels and red squirrels are good at is dealing with predators. They'll go up a tree and attack an owl and that sort of thing. So in forests, and in wildlife areas, the red squirrel will dominate, the fox squirrel will dominate. You won't see gray squirrels as much. Now, on the other hand, if you see gray squirrels on a college campus, that means either there's coyotes there or there's feral cats. So again, evolution tells you the truth, and our job is to understand. So in this case, when we do imaging with radiologic studies, so these are large scale, kind of the equivalent of satellite images. If we can identify certain habitats that we know in which there will be typically certain kinds of tumor cells. So, for example, if it's an area that's very poorly perfused, we would expect cells that are, hypoxic, that acidic, they have those kinds of capacities. So by understanding how the large scale environment selects for the small scale population, you can then estimate the subpopulations of the cancer. The problem is that it's really hard to do that. What we did was take MRI scans. So MRI scans are the same tissue is repeatedly interrogated with different sequences that are sensitive to different components of the tissue. Putting those together, we can generate a habitat. But this is technically very difficult because the sequences are never precisely the same. The spatial scales are often slightly different. For example, there can be a 3 millimeter slice thickness versus 5 millimeter, even 7 millimeters. So technically, there's a lot of things to overcome. What we have pleaded with the imaging companies to do is to develop a sequence that allows you to interrogate the same tissue with multiple sequences simultaneously, which in theory is possible. But that's been very slow to develop. So it's been a frustrating path, and this is sufficiently expensive that we need grants for that, and they have not been forthcoming.
Mike, any other questions?
No, a lot to think about it. I think this really this consilience of evolutionary kinds of considerations over the population and the sort of software aspects in the decision making of the collective and these physiological circuits. I think this is where in the end the solutions are going to come from. I think we really need to understand both sides of it.
It's funny, if you go back to the era when the human genome was being deciphered and if you read the literature, it sort of implies that this is pretty much it. This is the end of disease. I mean, I think that term was even used. And it's funny because we still live in the very genetic era where everything that's done is based on genetic measurements, and I think that it's a whole generation I think has been almost lost because there's been so much focus on that we've given up on many of the other things which are actually being developed in the 50s and 60s, but then just kind of got passed over by the genetic revolution. It's just so easy to put your sample in a molecular biology machine and generate data.
Yeah.
And I think there's a false sense that, all this data is going to tell us something, really great. An interesting analogy is to say, suppose you were a modern Darwin and you were on the Beagle with just $1 million worth of molecular biology machinery. And suppose the sailors tramped through the Galapagos Islands and they brought you back finch samples, and you ground them up and put them into your molecular biology machines and generated terabytes of data. Could that modern Darwin have written on the origin of species? And I think the answer is no. Although I've posed this to molecular biologists who always say, "Well, I think we could have done it." But the problem is that there's not a clear mapping from genotype to phenotype. So you would have to know from the gene that there's something about the beak, but you lack what you cannot see is the selection force, the morphology of the beak. I mean, what Darwin saw was something very straightforward and logical. The morphology of the beak and the morphology of the seed matched. And I think for all the data we generate from molecular data, I don't know if we've gotten a true sense of what's really going on. I mean, I think we've sort of missed we've missed the seeds, and in cancer at least we've not done the phenotype, the beak versus seed kind of understanding, and so we've got lots of genetic information, but it's not being built on a solid framework of sort of evolutionary first principles.
Yeah, I think your framing of where is evolution spending energy and how 1/3 of it is, say, even just ion channel transport, it just tells you a lot where perhaps we should be focusing our efforts on other things as well.
Yeah, follow the money. Yeah, I think evolution is trying to tell us something, and we've not been listening very well up until now.
Hi, everyone. If you wish to be notified of future podcast episodes and my writing on longevity and the frontiers of biology, please subscribe at livelongerworld.com. And if you're enjoying the show, please leave a rating on Apple, Spotify, or elsewhere. Thank you for listening, and I will see you next time.